Hello, today we're going to be discussing Helicobacter pylori, its implications, prevention, and treatment methods. First, we'll start off with an overview. We will introduce the topic, talk about how to identify the bacteria and its prevalence, move into pathogenesis, how the bacteria is successful at colonizing in the gut and causing disease. Then we'll move into prevention, some opportunities with probiotics for prevention and treatment options, and then we will move into species differences not only between Helicobacter species but also species of animals that can be infected with the bacteria. So first, as discussed, we will introduce the bacteria, talk about how to identify it and its structures, and how it's prevalent in our society. So here's an image of Helicobacter pylori in a colony in the human gut lining. It is a gram-negative bacteria that has a sort of spiral shape with flagella that help it do exactly what it does, colonize in the gut. Some clinical consequences of this colonization include gastritis, which is inflammation in the stomach lining, ulcerations, which are essentially open sores in the stomach lining, and gastric cancer. In fact, the first formally recognized bacterial carcinogen was Helicobacter pylori, which really just solidifies scientifically that this bacteria can cause cancer. So the mode of transmission of Helicobacter pylori is not fully understood, but it's likely some combination of human-human transmission and contamination of food and water sources. Because of this, 800,000 new cases of gastric cancer associated with H. pylori were diagnosed in 2018 alone, which is impressive for the bacteria because only a little bit over 1 million cases are diagnosed every year. The global prevalence is 43.1%, meaning that 43.1% of humans have Helicobacter pylori in their gut. As we can see in this graphic over to the side, it occurs in not only developing countries, but also developed countries, with up to 40% of developed countries like the UK, Australia, and France having antibodies for Helicobacter pylori. And we can see in countries like India, by age 60, almost 100% of their population has the antibodies for Helicobacter pylori. We see that in lower income countries where there is lower health coverage, there is a higher prevalence. And we also see the same thing with children. We can see in some of those developing countries that even before age 10, around 60% of their children have Helicobacter pylori antibodies in their gut. Next, we are going to talk about the pathogenesis of Helicobacter pylori and how it's successful at colonizing in the gut. This bacteria is a part of the etiology of many gastric diseases due to its unique and variable pathogenesis, so it is important to understand how it works so that we can treat it. So first, there's little competition in the gut because of the low pH and high acidity of the stomach. So the bacteria, Helicobacter pylori, doesn't have a lot of other species to compete with as far as space and resources go. So urease-positive bacteria are able to survive in very acidic environments by hydrolyzing urea into ammonia, which therefore raises the pH and makes it less volatile for the bacteria to live there. The flagella that this bacteria possess help it swim towards higher pHs so that they aren't being impacted by acidity again. And finally, the spiral shape helps it penetrate mucosal layers and has good hydrodynamics as pictured to the right when swimming through the gut and helping it eat into the stomach lining. So next, we'll discuss some of these consequences of successful colonization in the gut by Helicobacter pylori. So first, gastritis. When these bacteria are able to infiltrate the mucosal layers in the stomach lining, they cause inflammation, assisted by the immune system, and this is called gastritis. So inflammation in the stomach lining is called gastritis. And when this inflammation gets very severe, it will cause peptic ulcers, which are essentially just open sores in the stomach lining. You can kind of picture the bacteria eating away at the epithelium in the lining of the stomach. And then finally, gastric cancer. Geographical associations tell us that places where there are high incidences of gastric cancer, there is also a high incidence of Helicobacter pylori. In fact, 60 to 80% of gastric cancers are caused by Helicobacter pylori, which kind of reflects what we discussed earlier about how many cases are being diagnosed of gastric cancer that are associated with Helicobacter pylori. So the way that it does this is the bacteria produces reactive oxygen species, which cause damage to DNA in cells and trigger this cancer cascade where cells are being damaged, and when they grow back, they are growing back too quickly and causing overgrowth of cells. So because of the outcomes of gut colonization and other closely related species, prevention and treatment research is essential. So probiotics have been being studied for their ability to prevent colonization with bacteria like Helicobacter pylori for numerous amounts of reasons. So first, barrier enhancement. We know that our mucosal membranes and epithelial barriers are the first line of defense for our immune system to protect against pathogens. So probiotics enhance this by promoting mucus secretion, making it more difficult for bacteria to travel and penetrate through epithelium surfaces. And then they also upregulate tight junction proteins, which serves kind of the same purpose of making it difficult for bacteria to stick and colonize epithelium. Second, by secreting antimicrobial substances like lactic acid and short-chain fatty acids, probiotics can cause damage to bacterial species like H. pylori by inhibiting their urease activity, which, as we discussed before, is essential for their survival in the harsh conditions of the stomach. So if they cannot raise that pH, then they will not be able to live in the gut. And next, the probiotics, when taken, will compete for space and resources in the lining of the gut, especially adhesion to the gut lining. So in combination, all of these mechanisms of actions contribute to an improved inflammatory response against infection. So next we will discuss some treatment options because it is impacting some of our most vulnerable populations. Treatment is really important, and working with antimicrobial resistance growing in our communities is an important aspect to recognize when trying to treat an infection like this. Eradication rates for Helicobacter pylori do not consistently rise above 80% because of antimicrobial resistance. This is especially prevalent in patients who had failed previous eradication measures in place or improper adherence to therapy regimes. 50 to 100% of the population in developing countries or developed countries like America have antimicrobial resistance. This is sometimes due to use of antibiotics like metronidazole to treat parasitic infections, and also its use in gynecological therapies for women that increases this antimicrobial resistance. In the last 20 years, triple therapy has been used to treat H. pylori infections. So this included a proton pump inhibitor, clarithromycin, and then either amoxicillin or metronidazole. But since this eradication rate does not rise usually above 80%, they have had to resort to some new options for treatment. So this new treatment has been called quadruple treatment or quadruple therapy. It includes the use of 2 antibiotics, which are tetracycline and metronidazole, in combination with a proton pump inhibitor and bismuth, and this is used usually for 14 days. And increasing this length and dosage works by minimizing the impact of those metronidazole resistant strains. Specifically metronidazole, we see a lot of resistance to that antibiotic. So considering this high incidence of H. pylori combined with the high resistance levels and devastating potential outcomes, research considering new treatments and prevention with probiotics are key to protecting communities around the world. Finally, Helicobacter's discovery and pathogenesis related to gastric ulcers and cancer has sparked clinical interest in its impact in other species and how maybe they can mirror the disease for us or how it's impacting species for their health. So there are several different gastric Helicobacter species. These are the most relevant ones that we see. So H. pylori, which we've been discussing, colonizes the gut of humans and primates. It's not found in dogs, and it's very occasionally seen in cats, and has a low or zero potential for zoonosis. Next is H. felis, which is found in cats, dogs, and mice, and while it hasn't shown potential to cause harm in cats and dogs, there is a potential for harm in mice. And then finally we have H. musculae, which is prevalent in ferrets, and it causes the same things it causes in humans, gastritis, ulceration, and gastric cancer. And because ferrets share anatomical and physiological structures and processes with humans, this can be helpful for us to model the disease, to study more preventions, treatments, and transmission. So currently there is not a massive concern amongst veterinarians on how these Helicobacter species are impacting animals. We know that they live in the gut of most of these animals and don't cause harm. So it isn't a super massive concern. There also isn't a very high potential for zoonotic transmission. There is a small potential, but not anything that has caused alarm in that community. But something important to note is that, especially with the ferrets, which is really interesting, is that since they kind of model our stomachs, we can use them as animal models to test different antibiotics, to test preventions like those probiotics, and then again to kind of get down to the bottom of transmission and how exactly it's happening. I think that this is really interesting because I have had a run-in with Helicobacter pylori myself, and I think that knowing that there are options out there to prevent it are important for everybody, because nobody wants to be caught up in a situation where they have resistance because of other things they've taken in the past, and that we can't treat it one day. So I think it's important for us as up-and-coming scientists to understand this and why it's important to our society. And here are my image links. Thank you guys so much for watching.